Hello, we are Group 14. We'll explain about co-stimulatory molecules in terms of its definition, a biological law, and examples. These three cells, called antigen-presenting cells, capture antigens when they recognize them. Macrophages phagocytose pathogens non-specifically. Dendritic cells work as the main character of APCs. They capture and process antigens, then present antigens. B cells work as a kind of APCs, produce antibodies, also become memory B cells to memorize antigens for future immune system. APCs have a kind of identity code called MHC class II molecules. They display it with antigens. First, we'll talk about the definition. Co-stimulatory molecules are a group of cell surface proteins that are essential for effective immune responses. They are seen on lymphocytes, such as T cells and B cells and antigen-presenting cells. Next, about a biological law. Let's look at it from the point of entry of pathogens into the body. When pathogens enter the body, the following occurs in the body. When antigen-presenting cells specifically recognize a pathogen, co-stimulatory molecules are expressed on the cell surface. Antigen-presenting cells expressing co-stimulatory molecules bind specifically to co-stimulatory molecules on the surface of naive T cells, a type of lymphocytes. T cells also have T cell receptor on their surface, which recognize antigens presenting with MHC on APCs. Both cells are stimulated by the occurrence of co-stimulation. In addition to this co-stimulation, antigen-presenting cells transmit antigen information, and cytokines cause naive T cells to differentiate into effector T cells. Effector T cells regulate various immune cell functions. There are some kinds of T cells, such as Th1, Th2, Th17, and regulatory T cell. In short, naive T cells require three steps for it to be activated: interaction between MHC and TCR, co-stimulation, and cytokine boost. While antigen presenting is known as signal 1, co-stimulation is called signal 2, as well as cytokine boost is signal 3. Activation of T cells without co-stimulation in general leads to unresponsiveness of T cells and apoptosis of T cells. Last, about example. We'll show some examples of co-stimulatory molecules. One of good examples of co-stimulators might be CD8086 or B7.1 and B7.2 and CD28. Antigen-presenting cells like dendritic cells express CD8086 on the surface, and the molecules link to CD28 on naive T cells. These co-stimulatory molecules cause co-stimulation between APCs and naive T lymphocytes, leading to activating them to effector T cells. The other example is the link between CD40 and CD40 ligand. APCs express CD40 on the surface. CD40 ligand exists on the surface of CD8+ T cells. Following CD8086 and CD28, co-stimulation by CD40 and CD40 ligand fully activates CD8+ T cells. Thank you for watching.